Are we ready to roll? All right. So we have a very small in-house audience this year, and I think that's because of the amazing success of our online streaming. Even the free babysitting wasn't enough to get parents here today. But for those of you who are here in person, we're delighted to have you here. And for those of you who have joined us online, we're equally delighted to have you joining us. The format for those of you who haven't been here before is that we're going to have speakers throughout the morning. Then there's going to be a question and answer period with a break for coffee and a photograph of the people who are here live. We can't get you online people. The way we do the questions is that we're going to have someone running around with index cards, and you're going to write your questions down, and then we'll have all the speakers come up here and triage the questions to the most appropriate person, and then you'll be able to go ahead and get your questions answered that way. And so for the people online, there'll be someone manning that and taking your questions as well. So here is our dedicated group of researchers at the Kawasaki Disease Research Center. Not everyone is here, but it's a big part of the group. And we have a very comprehensive research program here at UCSD and Rady Children's Hospital that includes both clinical research as well as bench research. By bench research, I mean things that we do with the samples that we take from your child during their acute and convalescent disease, and the progress that we're making in understanding more at the molecular level of what Kawasaki is and how this disease operates. On the clinical research side, we have all of the clinical trials that involve new medications, improvements, particularly for children who have early changes in their coronary arteries, and we're going to be talking a little bit more about that later. The data for how many patients we've seen at Rady Children's Hospital is shown here, date of onset between 2004 and 2018. You can see that 2018 was a very low year for us, but in general, the line is headed up. And we're going to share with you some data about what 2019 looked like, which was a very big change. Here you see those same numbers broken down by the race ethnicity of our patient populations. And the green bars stand out very prominently, and those are our Asian children. And we've lumped everybody together here. We have a project ongoing now specifically describing our Vietnamese Asian children. And we'll be presenting that next year when we have those data fully analyzed. We actually had a medical student go over to Vietnam and work with a group there so we could learn more about Vietnamese children and Kawasaki disease. So you can see that in any year you want to look at, our Asian children are overrepresented, but African Americans make up another very prominent group. And of course, this speaks to the genetic susceptibility that underlies why your child got Kawasaki disease. And that's an important part of the piece that we're trying to unravel in our laboratory. This is the mix of what Asian means here in San Diego. And you can see that the Filipino population is very prominently accounted for here. And in addition, we have the Southeast Asian population, largely Vietnamese, and Chinese population as well. But that's what our Rady Children's Hospital KD patients look like. So here's what happened this winter, and for those of you who live here, you know that it was a very unusual winter, and we had very unusual clustering of patients. So a couple of things to notice. What you see here is that we've got red dots for the date of onset of every patient that we diagnose. Now, we may meet the family and meet the patient on day 5, 6, or 7, but this is actually the date on which they had their onset. And you can see that in February, we had these massive clusters of cases where there were many children. Even in May, we had this, where there were many children with their onset on the same day. We don't think this is a coincidence. This is real. This means that there was an exposure in our county at that time, and these genetically susceptible children were affected. I also want to point out the missed KD patients. These are the gray dots here. And we're sad about those gray dots. We would like to eliminate those gray dots because these are the children who were diagnosed so late in the course of their illness that they were really beyond the point in time when IVIG was going to be helpful to them. Fortunately, this year, but this doesn't always happen, all of those gray dots represent children who are not genetically susceptible to the cardiac problems children. So we think that getting the heart complications is a genetic subset of the total population of Kawasaki disease patients. But we would like those gray dots to go away. Dr. Tremblay and I reach out to doctors in the community. Our emergency room doctors are incredibly well educated about this disease. And we try very, very hard to not have these children missed. But it's still happening, even in a community like ours where there's, I think, pretty enormous awareness of the disease. So still room for improvement. Well, this is what our winter was like for those of you who were here. And we don't think this is a coincidence either. It was an anomalous climate event that we had with these atmospheric rivers swooping into the county. We then had these high pressure events that followed it. It's a very unusual climatology that was associated with these patients. Here are our total numbers up until the middle of October. So you can see that whereas we usually say, parents say, how many Kawasaki disease patients do you see every year? At Rady Children's Hospital, we care for about 80 to 100 each year. But we're not at the end of the year yet. In fact, November, December tends to be the beginning of our big season. And you can see that we have already hit 96 patients this year. And you can see how busy we were in the winter months here. Again, these are the missed KD patients. We would like that number to go to 0. And this gives you an idea of the distribution of the months when we saw patients. So we are very actively studying the climate and what its effects and its interactions may be that create the exposure that triggers Kawasaki disease. And I'll be talking about that a little bit later in one of the other talks this morning. So of our roughly 800 patients, you can see that about a quarter of them developed criteria for calling them having a coronary artery aneurysm, and our Asian children were overrepresented in that group. So something about the genetics of our Asian children tends to make them more susceptible to the heart damage. Now this is from a handout that we have for parents. It's online. You can get it, and it explains how to understand the echocardiogram results that we do on your children and what a Z-score is. So we now call an aneurysm anybody who has a Z-score, which is basically the internal diameter of this blood vessel, and then it's normalized for body surface area, and we express it on this curve here as standard deviations from the mean. So if your Z-score is 0, you're right there. And if your Z-score is greater than 2.5, we consider that to be an aneurysm, which may resolve quickly. It may have many different paths. And in the second session today, Dr. Gordon is going to be talking about what those different paths might be. So how can we prevent aneurysms? In terms of the long-term outcome, long-term complications of the disease, we really think that our children who have had always normal echoes, that right now there's no cardiovascular signal from that population that they have a problem. However, the children with aneurysms, it's a different story, and you'll be hearing more about that later in the talk. So how can we head that off at the pass? All of the effort right now is being focused on intensifying therapy right at the beginning. We feel that there's an urgency to giving additional treatment to our Kawasaki patients who have an initial abnormal echo. And there's been a lot of discussion in the literature, and this is a paper where we contributed all of our data from Rady Children's Hospital to the group in Boston and Montreal, and we joined together to do an analysis of different practice approaches. If you are a Kawasaki disease patient in Boston, and you have an abnormal echo, a Z-score greater than 2.5. You are treated with a second dose of IVIG and an oral regimen of steroids that goes on for 3 to 5 weeks. If you are in San Diego, you get a single dose of infliximab, which is a molecule that modifies the immune response. And if you are in Montreal, you get nothing in addition. And all of these are legitimate approaches based on the current data from the American Heart Association guidelines. So no one was doing anything wrong, and no one was doing anything necessarily right, but these were different approaches in a protocol-driven way that we approach these patients. And here's the outcome. And again, this is published data. If you were in Montreal and you had an initial abnormal echo, the chances of your Z-score increasing by 1 standard deviation, so getting even worse, that was about 60%. Whereas if you were in Boston or San Diego, your outcome was roughly the same. So we think that this, although it's what we call retrospective analysis of data, that's always hypothesis generating, is not the end of the story. It needs to be studied prospectively, but here we have evidence that intensifying the initial therapy for our children is a good thing and should be done. And the choice of what you use, we need to study that. It does look like these two approaches are pretty much equivalent. So how do we know which patients actually need this intensification of treatment? Well, I told you that we could estimate that from the initial echo, and 80% of the children who will go on to have aneurysms have an abnormal first echo. So we know right then and there at least 80% of the children. So we teamed up again with the Boston group, where we do a lot of collaborative research. Collaboration is very important in Kawasaki disease because numbers are generally small, so you have to join together with other centers. And in Boston, Mary Beth Thon, shown here, worked with the statisticians to come up with an analysis of our data and Boston Children's Hospital data to figure out a risk score. And after a lot of work with the data sets, they came up with a scoring system where you got 2 points. This is a scoring system where you want to be 0, like you don't win if you have a high score. You want to have a low score. And the echo being abnormal, greater than a Z-score greater than 2 gives you 2 points, 1 point for being under 6 months, 1 point for being Asian, and 1 point for having a very elevated level of a marker that we call CRP. So this is what happens if you have those various scores, and you can see that if you have a score of 3 or greater, this was done twice, so first a development cohort and then a validation cohort. So altogether we had over 1,000 patients that we pulled together from our 2 sites to do this research. And you can see that 40% of the children who had scores of 3 or greater in this group had developed aneurysms. And in the second testing of this scoring system, that was even more, so roughly half of the children. So that seemed like a pretty good scoring system. This is another way to look at it using odds ratios. And so if you were in the moderate risk group with a score of 2, you had a 5-fold increased risk of having aneurysms. And if you look at the results from the validation cohort, so again, an independent group of patients, you can see that your relative risk, meaning what's the chance that you're going to have end up with aneurysms, was very high with this risk score. So we're looking at this now as something that could be used for a national trial. And here are the clinical trials that are ongoing right now. Thank you to all the parents out here who participated in these trials. For these trials, you are only invited to participate if your child had an abnormal first echo. And you can see that we're looking at a class of statins. This is a drug that we've now published the first study from that, and I'll show you that in a minute. Anakinra is a way of modulating the immune system and reducing inflammation. And now a trial using both of these. And all of these are being run by Audrey Tremblay, who is our clinical trial specialist in our research group. We have an additional trial for children with giant aneurysms where we're testing a new approach to blood thinners or anticoagulants. So it was a huge triumph in getting the project across the line to get this very large study in terms of all the components to it. It wasn't a large number of patients, it was a little over 30 patients, but a huge effort to get this study here at the University of Colorado. Again, our Katie Joan worked with us, but Audrey led this project, and it basically established that the use of statins, and I looked this up online, and apparently 35 million Americans take a statin every day, but these are adults, and they're largely taking it because of lipid issues. It turns out that statins are wonderful drugs in many other ways and work to reduce inflammation. And we think it's more along those lines that it's working in Kawasaki disease. And it was important also to make sure that we were doing no harm to our children. So a phase 1 trial is really about safety. And so Chisato Shimizu, who runs our molecular programs in the lab, was able to look at this molecule here, which is your brain cholesterol. So cholesterol is good for you. It's not bad for you. It's part of your brain. And growing children are growing their brains, and we wanted to make sure that we weren't harming them by giving them something that was potentially lowering their cholesterol, not because that was what we wanted to do. It was almost a side effect of the drug. And what she showed is that whether you were treated with a statin or without a statin, your levels were the same, and we didn't need to worry about that safety issue. So the dose for your children was established through this trial, and the safety of this drug was established through this trial. So very important landmarks along the way in pediatric research. And now, Dr. Tremblay, along with Kevin Friedman in Boston, is planning a national trial. They've been awarded a grant by the National Institutes of Health to spend a whole year trying to plan a novel trial design where we can intensify therapy for the children who have early signs of aneurysms, or maybe we use the scoring system that I showed you. And this will be something that will be rolled out across the United States, and we hope to test different approaches to intensifying therapy. We do have the Kid Care trial open, and thank you to all the parents who participated in this. This is a different study that is just for children who have recurrent fever after their first IVIG infusion. And their bodies are telling us they need a little more anti-inflammatory therapy. And we're comparing a second IVIG dose versus infliximab, which is both of those standard treatments can be used at various hospitals across the United States, but now we're organizing it to compare those two. And finally, yay, we're across the finish line thanks to all of you here in this room and online and across the globe, really, with other countries participating as well, where we made the match for the $2.5 million Macklin Foundation matching grant, challenge grant. And it's really thanks to this lovely person here who's with us today. And if any of you have the desire to continue to contribute to our research, we are always in need, and we welcome all of those contributions. Amy Weeks is the Senior Development Officer at the Rady Children's Hospital Foundation, and really single-handedly worked with our parents to make the philanthropy a real thing, to make the match and get us over that finish line. So with those $5 million, we supported all of the people that you saw in that first slide, and are continuing to work to make this world a better place for our health sectors and children. So thank you, and we are moving on now to the next speaker. So Dr. Tremblay is going to come up and tell us about the work that she's doing in Latin America that is very unique. It's really a one-of-a-kind project, and we hope making a difference for all of our neighbors to the south. Thank you, Audrey. Thank you.
an absolute pleasure to be here and be able to share with you guys the work that we've been doing. And I'm going to actually go back quickly just to let you guys know what Vega Latina stands for. It's I am a Cuban American, first generation, speaking Spanish at home growing up. And after learning a lot about KD here, I started to ask some questions about what was going on in Latin America. I was invited to give a talk at the Congress in Ensenada, Mexico in 2009. I decided I would just Google Kawasaki disease in Latin America, and I came up with one paper. And I thought, well, that can't possibly be true. And so then I looked up KD in Mexico, because I was going to Ensenada, and I found 73 cases in 30 years, which is a gross underreporting for 32 million children in Mexico. We have the good fortune that over the years, since that talk about 10 years ago, of meeting a number of colleagues along the way, there's a very active group now throughout all of Latin America publishing Kawasaki disease and really trying to improve the lives of children within Latin America. And I want to share our role in doing that with you here as well today. So in terms of when I was in residency in pediatrics in Los Angeles, I wanted to do rotation abroad. And my advisor at the time said, well, you speak Spanish fluently, you should go to Latin America. I have a contact in Costa Rica. So I went and I studied medical school in English, not in Spanish. The first day I couldn't even find what a medical record was. I did not know that there was even a medical record in Spanish. I learned quickly over those 6 weeks of being there, became fluent in medical Spanish while I was there, and made a really amazing contact who was this lovely person who put Ávila in Costa Rica, Hospital Nacional de Niños, in the hospital for children there. And it turns out that our relationship grew. I recommend to every student that if you want to do something, reach out to someone that you see as a mentor and offer to help them out. I actually offered to help her write a paper that a medical student had failed writing a couple years ago. It was one of my first publications, absolutely nothing to do with Kawasaki disease. But when I was reaching out to Latin America, she was the first person I thought of. Because I'd actually helped her out and been there with her through that process and gotten that paper over the finish line, she immediately called me back and said, well, I'm not the Kawasaki person here, but this person is. It was Rolando Ulloa, who is a pediatric infectious disease specialist, who I actually never met. He already worked there at the hospital, but he was up in Canada doing a fellowship while I was down in Costa Rica. So we became close friends. He was, of course, plugged into the pediatric infectious disease network in all of Latin America, and we quickly took this running and really have made a wonderful stride over the last 10 years. So one of the things that Rolando shared with me early on was between 2000 and 2010, there was a lack of reporting in Central America. That included Costa Rica. You see there that there were a report of 8 kids in Costa Rica and only a handful of trials. So it was still being grossly underreported, and we needed to do something about that. So we actually made a pitch to the Latin American Society of Pediatric Infectious Diseases, known as SIPE, and we were invited in 2011 to Punta Cana, the Dominican Republic, to give a talk. And I naively thought that there would be 5 people in the room. So I showed up with my slides in Spanish after having lots of people. I looked at them, I looked at them. Great aunt who used to actually teach Spanish literature looked at them and make sure my accents were correct and everything. I got up there into a hall with 500 people in the audience. Physicians that were intrigued and interested. We're seeing a lot of patients with Kawasaki disease. Had some amazing questions, and we really wanted to be able to have a standard created throughout all of Latin America. And we're doing the best with a reasonable limited setting for most of them. Scary part right after that was that I actually had a session where I had to do question and answer in very rapid Spanish, and I know I speak Spanish quickly because I'm Cuban. When you've got people from 20 countries with 20 different accents asking you questions in medical Spanish, I promise you that was the most intimidating thing I've done in my life when it comes to my career. But it was well worth it. From there we went on, and now every 2 years when SIPA happens, we have a spot. We have our own symposium that lasts about an hour on Kawasaki disease specifically, and it's been really able to grow our network. So what have we been able to do? We have 150 investigators across 50 hospitals in all 20 Latin American countries, including my family's native country of Cuba. Progress thus far, we decided we needed a logo. So we actually went back to our investigators and we said, What would you like to see in that logo? What becomes part of it? We had a great graphic artist that was actually one of our colleagues in the group, and he created a number of different designs. It was approved by, kind of the whole group in essence, and that's become our logos instead. We also decided that we needed to prospectively and retrospectively collect data on Kawasaki patients to really have an idea of what was going on. So our own group here took that task on using RedCap, which is a research electronic database that we've been using here for years in collecting information from your own children. We used that and we translated it to Spanish and had these 150 investigators put in information about their Kawasaki patients. And so far, that's nearly 2,000 Kawasaki patients that we've now collected over a 10-year period. So an amazing, rich data repository. This took a huge amount of effort from our colleagues, from our collaborators, from the folks here at UCSD, supported in so much by learning what we have been able to do over the years here with your own kids' data as well. And really being able to capture things like, what's the age of the child? Do they have all kinds of signs of Kawasaki disease? Is their heart abnormal? How many echoes did they get? Did they get treatment? If they didn't get treatment, why didn't they get treatment? So we're really able to tell the world about Kawasaki disease in Latin America. It was the first book that's ever been published in Spanish about Kawasaki disease. I can't take absolute full credit as the Latin American network, but I can say that I'm very thrilled to have the two authors of this book as part of our network. And I had the great privilege of writing the foreword and being able to read through it. Let me tell you, that stepped up my Spanish. My Spanish and medical Spanish, definitely. So we now have a book that tells Latin America about Kawasaki disease. In addition to the weekend presentations, we've also been privileged at the International Kawasaki Disease Symposium that happens every 3 years. For the last several times, we've been able to actually feature our network, have speakers from our network, and have Latin America as on the world stage discussing what's happening there in Kawasaki disease. So we now have had over 100 presentations worldwide and really trying to spread the word about KD. Publications, we've also been able to get the word out. So one of the things that I immediately found out is that very little was being published, and when it was being published, it was actually only being published in Spanish, and it was not making it into PubMed or Cielo. It was not readily available, so nobody knew that it was being published. So we made a big deal about making sure that we get things published, sometimes bilingual, sometimes monolingual English. We also push for things because Brazil's part of our network, just writing things also translated properly into Portuguese, and to have as much indexed in PubMed as possible so that it's readily available around the globe. And then we've also had a number of other publications that have made their way out, and it's taken quite an effort to get this data repository of nearly 2,000 patients. We're just finally at the stage where I can say that after all the data cleaning and all the data collection, that we're now at the point of really being able to use that network and use those data to be able to publish even more and tell the world about what's going on in Latin America. Early lessons learned. I'm going to go through just a few things to highlight, kind of point out things that are similar and different than what happens around the rest of the globe. Illustration diagnosis. It turns out the children in Latin America are generally not being diagnosed all too late. There's still the missed cases, just like there is here. It's kind of those same inherent issues. For the most part, we found that the data this wasn't all that dramatically late in terms of the time of diagnosis, sometime in the first week or so. Distribution of males and females, where we usually see about 2/3 males and 1/3 females, same throughout Latin America. Antibiotic use. So if your child comes in with fever and a rash, in Latin America, there's a lot of use of antibiotics. And so over 90% of our kids there are being treated with antibiotics from the get-go, whereas that seems to be a lot less in other parts of the world. And now with antimicrobial resistance increasing, it becomes an ever-urgent issue. And so we're really trying to be able to educate our primary providers throughout Latin America about the use of antibiotics and when it can and when it shouldn't be used and when you can be thinking about KD. In terms of infants with Kawasaki disease coronary damage, this is super interesting. I shared some data last year about children less than 6 months of age and the fact that even if they're treated in the first 10 days of illness, half of them get the heart disease. The story is very similar throughout Latin America. There's something about our infants where they seem to be predisposed to having damage very early on and quite severely. It's something that we need to just be able to get the information out there. Because now what we've been able to do through those publications and letting people know about the little babies having the mad heart disease is to get people to think about Kawasaki disease. Once they reach Dr. Burns and I, we're able to take care of them. We don't know what's out there in the community. So it's important for our providers throughout the world to know that infants are especially at risk for having severe heart disease. Use of serial echocardiograms. Okay, so the standard is you not only get one echo, but you get more than one echo. And what we found in a lot of our data in our patients is that getting that second echo can be really tough. And it can be tough because of the resources, it can be tough because you don't know who they followed up with, because of loss to follow-up. And so this is a big push, and really pushed me as an investigator to say, well, who do we not connect with? Well, we're not really engaging our pediatric cardiologists. That's been a big part of this network, is not just to keep it with the infectious disease people, that's how I'm trained, but to also reach out to our cardiologists and say, hey, it's super important that when that person, when that physician caring for that kidney patient asks you for a second echo, don't put up the fight, do a second echo because we need it, right? When they go home, get a follow-up echo. These are the kinds of important follow-up issues that we're trying to address. And finally, availability of standard and novel Kawasaki treatments. So time and time again, I've been amazed by two things that seem to be polar opposites. One, that in fact in many hospitals, IPIG is readily available. But then there's also those areas where it's absolutely not, where you have to be, having an argument with people that run your hospital or aren't able to treat that patient with IVIG, or you have to transfer them out. And then as the clinical trials of the group, I'm always looking at the new thing that can be used. But if I go and give a lecture on the new thing that can be used, but that new thing isn't available, it's really kind of useless. So it makes us really cognizant about what's going on at a very local level, and to be able to really be able to come up with new treatments that maybe are cheaper, that maybe are easier to get to, or easier to get than even IVIG. So that brings me to this last point, which is just some beautiful work that's been done by one of our collaborators, Dr. Alessandra Franco. She's an immunologist. And she has been looking at, is there a way that we can be a little bit smarter about understanding how IVIG works? And really being able to maybe come up with something cheaper, something better that can be used, especially in those resource-limited settings. So let me walk you through this. Up there on your top left-hand corner, you have IVIG. Looks like a little Y, that's the antibody that then gets put into the cell, and is chopped up by those little scissors. Okay, so it's getting processed. And then it goes out the other side there, in the middle of the screen, and it comes out in these little itty-bitty parts, okay? And these peptides come out, and then they are presented to the cells in your body. And this is what happens and how IVIG is processed. So Dr. Franco asked the question of, is it possible that we can just be making those little itty-bitty parts, right? And IVIG is a human blood product. It's one of the most expensive medications we have in our pharmacy, and it also runs the risk of being a human blood product. And as safe as it is, that's what it is at the end of the day. So can we maybe process, just take those little itty-bitty green parts, make them in the lab, synthesize them, make it cheaper, maybe it's even safer because it's not a human blood product. Certainly easier to get. And so that's what she's working on in her lab, and trying to see if she can get those peptides to work. And if maybe that will be at some point a solution, an additional form of therapy for our treatment, for our patients, and maybe even something for areas where they have trouble getting IVIG or it's too expensive to get. So with that, a very big thank you, and Muchisimas gracias to all our collaborators. This is an amazing group effort. Certainly haven't done this. Rolando and I are indebted to everyone. Our emails are there. If anyone that's listening in Latin America wants to be part of our red, absolutely. Anyone who wants to learn something from the data, it's there. It's waiting for you to look at it and write that amazing paper to educate the world. So again, thank you very much for the opportunity to share this with you.
Thank you, Audrey. Our next speaker is going to really switch it up here. We've been talking about children and humans, and now we're going to switch to animal models and fish. This is an exciting new collaboration. This is part of why it's wonderful to be on a major university campus with smart, young, talented scientists all around us, and our excitement to be able to reach out to these people working in very different areas, but who may have something very important to contribute to understanding how Kawasaki disease works. So Crescent and Madigan, thank you so much for coming this morning.
Good morning. Thank you so much to the organizers for giving me this opportunity to tell you a little bit about my work. So I'm a professor of molecular biology at UC San Diego. I just started my lab. I've only been here for about a year. And I'm a PhD, not a clinician, right? So I work on basic biological processes. And my lab is focused on understanding inflammation using a new animal model that's something that's been increasingly popular in science over the past maybe 20 years, but hasn't really been applied to a disease like Kawasaki disease before. So when I first came to UC San Diego to start my lab, I was watching news one night and I saw an interview with Dr. Burns on the TV. And I got so excited because when I was getting my PhD in Boston, Kawasaki disease was one of the things we learned about. And I thought the biology behind it, back then, I thought the biology behind it was fascinating. And so I was excited to find out that there was a clinical research center here. And I reached out to ask if there was anything the zebrafish could do to contribute. And so, the zebrafish have been working hard. And I'm going to show you a little bit about what we're doing to try to make a zebrafish model for Kawasaki disease. So, first of all, this may be obvious to some people, but just to make sure we're all working from the same knowledge base, when scientists want to understand how a disease works, one of the things we do is use animal models. And the reason that we do that is because diseases are so complex and involve so many different cell types working together that it's very difficult to understand the disease process fully if you're just working on cells growing in a dish. And so many people will use the animal model you've probably heard most about is the mouse, right? Scientists use mice. And mice are fantastic models and have really advanced our understanding of the biological processes behind diseases. But there's some things that the mice can't tell us. So basically, the reason that people started using zebrafish, which we're showing right here, to understand diseases is that with mice, they're very difficult to see inside of. So if there's a disease happening inside of the mouse, for example, in the heart, one of the best ways to see the heart in the mouse is to cut it open, right? But of course, that's the end of that mouse's life. And so what we can't do by surgically exposing the heart is watch how the disease progresses over many days or many weeks. And that's because the mouse is opaque, right? You can't see inside of it. If I were to shine light onto it, the light would be totally blocked. So zebrafish, when they're larval, so when the zebrafish are babies, they're completely transparent. And so that means that if we shine a light onto the fish to look at the disease process, we can see everything without having to do any surgery on the fish. And that means that the fish stays alive and we can watch how things change over time. And that's a really powerful piece of information that it's hard to get at sometimes with mice. And so that's why my lab uses zebrafish. And my particular interest with zebrafish is looking at how infections cause inflammation in the brain. And we're looking at how blood vessels in the brain get inflamed during infection. And so that's quite complementary to some of the work that Dr. Burns is doing, looking at how the blood vessels become inflamed and damaged in Kawasaki disease. So to give you an example of some of the things we can do, so this is a 2-day-old zebrafish larva. So it hatches at about 2 days old out of the eggshell, and it develops outside of the mother. And because it's transparent, we can see every part of this animal. So right here in the tail are the blood vessels. You can see this movie we've used genetic approaches to label every single blood vessel in red. And you can watch the behavior of every single blood cell as it moves through the blood vessels. There's also a lot of work being done to understand heart function in the zebrafish. So the heart in a 2-day-old animal is right here. And you can see in this video, the valves of the heart have been labeled in fluorescent green. And so just by shining light onto this animal, we can watch every movement of this heart. We can see if there's inflammation in the walls. And we can watch how those things change over time just by taking a movie. So some of the reasons that people like zebrafish are because the genes in the zebrafish are quite similar to humans. There's about 70% of the genes in humans are represented in zebrafish. There's really lovely genetic tools in the zebrafish. So in most animal models, if you identify a gene that you think is contributing to the disease, a test that you might do to prove that that gene is doing what you think it is you might delete it. Or you could add in a new gene, right? And so because the zebrafish are so small and so simple to work with, there's amazing genetic techniques that have been developed for use in the fish. And that's what allows us to make movies like this, right? So we have to use genetics to make things different colors in the animal. And so it's really this combination of genetics with live imaging inside of an intact living animal that allows us to get some of this really remarkable information. And then finally, when it comes to what we're going to do with these animals to contribute to the clinical research that's being done on Kawasaki disease, is that the zebrafish are really, really small when they're babies. So this animal is about 1 millimeter long. So it fits on, if you look at a penny, it fits right on top of the tie that Lincoln is wearing on a penny. So that's how small they are. They're really tiny, right? And also, there's a lot of them. So one female zebrafish lays about 300 eggs every week. And so I have, I think, something like 10,000 animals in my lab. And so that means that we can make a huge number of zebrafish larvae. And because they're so small and so numerous, that means we can do screens in whole animals. So a screen is where you don't know what you're looking for in terms of the molecule, but you know what you want to see. So what we want to see here would be limiting inflammation. And so we can do an unbiased screen in zebrafish because there's so many of them and they're so tiny. And so that could be something that we could use to identify new potential targets for patient therapies. So some of the work that my lab does, I like to show this video to sort of illustrate what's possible, is we're really interested in looking at how the function of the brain is changed by infection. And so zebrafish have really been leading the charge in this area of research. So this gray shape is the intact brain of a living animal. And because the fish is transparent, you can see everything that's going on in the brain at the same time. So this fish has been genetically altered so that every time a neuron fires, you see that flash of light. And so this animal is lying on a glass dish, and a brine shrimp swims in front of it. And that's what all those flashes were. The fish got excited and wanted to hunt down the brine shrimp. And so this is the only animal where we can do this kind of whole brain imaging to understand things like how a fish might hunt or what kinds of neurological injuries might happen during an infection. So this is the kind of work that my lab does. But turning to Kawasaki disease, so one of the first experiments that I did in the zebrafish to contribute to this research was to look at some of these inflammatory cells that we think are important to Kawasaki disease. And so these particular cells we're looking at here are called neutrophils. So we took fish and we treated them with saline, which is the control, or we treated them with a molecule that we think could trigger Kawasaki disease. And we saw in these animals activation of these inflammatory neutrophils. So here's a picture of the injection site in these fish with saline. And then here's a picture of the same site where we inject this Kawasaki disease molecule. And you can see that you get all these fluorescent neutrophils attracted to the site where we did the injection, suggesting that in zebrafish, there's an inflammatory response to this model Kawasaki disease trigger. So moving forward, so last night, we got some new data hot off the presses that I actually haven't even showed Dr. Burns yet. So it's the first time, it's debuting right now. This particular experiment was looking at these biomarkers for Kawasaki disease. So Dr. Burns and others have identified certain genes that change in Kawasaki disease patients. And so here's a list of some of those genes, and you can see that some of them go down and some of them go up. And so what we wanted to know in zebrafish is, do we see that same change as we see in the patients? And so last night, one of the things we found is that the zebrafish respond the same way as humans with this set of 4 genes. Now, this is the first time we've done this experiment, so we need to make sure that this repeats. We need to find other ways to confirm this data before we can be sure that this is true. What this data suggests are that this zebrafish model of Kawasaki disease really looks in many ways a lot like what's happening in the patients. And so this is encouraging and suggests that we'll be able to use this model to understand the basic biology of what's happening here. So this is one of the things we wanted to do. This is on our list of experiments, is look at whether these biomarkers in fish are changed in the same way that they are in human patients. We also plan to take some of these aerosols that are associated with Kawasaki disease that Dr. Burns and others have identified and use them to treat the fish and then see what happens to these genes. Are they regulated in the same way that they are in patients? Another idea that we've discussed is making avatars using the fish for Kawasaki patients. So we know that there's this genetic association with Kawasaki disease, right? There's certain differences in DNA that can predispose you to catching this disease. And so one of the things that fish have been used for is we can take the patient DNA, put it into the fish, and then ask, how does this affect the disease process, to learn more about what those genetic changes are doing in humans. And then finally, as I mentioned, we'd like to use the screening technology in the zebrafish to identify new targets for patient therapies. So the way this works is that I told you that we have a lot of these zebrafish, right? We have many, many thousands of them. And they're very small. So what that means is they will fit into a 96-well plate. So a 96-well plate is about the size of your cell phone, and it literally has 96 different wells. And we can put one fish into each well of the plate, and then we can take a library of drugs that have already been FDA approved, or different genetic knockouts, and add a different one to each individual well, and then ask, how does this change the disease process? Does it make it better? Does it make it worse? And so this is potentially a way that others have used this approach to identify new therapies for other diseases. And so my hope is that we can do something similar for Kawasaki disease. All right, so that's it for me. Thank you so much for your attention.
So you all can say that you heard it first here, cutting edge, amazing research. Thank you, Cressida, GoFish. That was so exciting. So just to be clear, what Cressida is injecting, and actually Greg Chin's daughter from the Kawasaki Disease Foundation was a guest student scientist in Cressida's lab this summer and started some of this work there. But what was being injected into those fish is a cell wall extract of the bacteria that's used for a quote-unquote standard model in mice that induces inflammation around the heart and the arteries that looks like Kawasaki disease. We don't actually think that that thing causes the Kawasaki disease in the children, but it's a way of creating very similar kinds of inflammation. So now we're going to talk a little bit about what does cause inflammation in Kawasaki disease patients, and what should we be giving to Cressida to put into her zebrafish to test. So this is the question that's centered to all of our minds about Kawasaki disease. It's front and center all the time for us at the KD Research Center is what is it that causes this disease? So we try to get clues from observing our patients, and we know that a large subset of patients come in with a swollen lymph node, and these lymph node chains here, which are shown here in a CT scan, so it's this big lump of tissue here. You can see it's not so much over here, but this big lump of tissue here. And those lymph nodes are like filters for the blood, and they importantly filter and enlarge when in a situation of infection or just being challenged by proteins or other molecules that are foreign to the body. So if you get a big dose of that into your mouth or upper respiratory tract, you're going to see these nodes enlarge. And if we use the CT scanner in a different angle, we can also see that there's an inflammation reaction here. This tissue should be lying flat against the vertebral column here. These are your vertebrae. And there shouldn't be this big space here of tissue that's very unhappy and inflamed. But this is from a Kawasaki disease patient. And then our ENT surgeons collaborated with us and used a fiber optic, what we call a spaghetti scope, it's very small, to look down at the vocal folds on our patients. And these structures should be very, very narrow and thin. And many of you may have noticed as parents that your child was hoarse during their acute Kawasaki disease. And that's because they have this massive swelling here. We always learn from our colleagues, and we only recently reported this within the last 10 years, and people have been seeing Kawasaki disease patients for a long time, but assumed that they were hoarse because they were crying. And it took our ENT surgeons to explain to us that actually children use different muscles when they cry, and you can't get hoarse from crying. So hoarseness is actually a pathologic change. And we used all of this information and these observations from our patients to come to the idea that these children are breathing something in. And whatever they are breathing in, that is what triggered this crazy immune response that they had. So I've just come from the infectious disease meetings in Washington, D.C., where Ann Rowley and the team from Northwestern University have discovered a new virus, and we're quite confident, the data seems very convincing that they have discovered a new virus, and they think that this virus may be related to the cause of Kawasaki disease. I think it's fair to say that everyone is waiting for more experiments, waiting to see what the future will hold in terms of more information. I think there's a lot of skepticism among the infectious disease community just because there are many features of Kawasaki disease that to us don't fit very well with it being triggered by a virus. But this is, it's always exciting to discover something new, and we certainly congratulate the group in Chicago for the work on discovering a new virus. I wanted to share this with you because this is something that's sort of gone out across the parent network, and you may have already heard about this. I would just say that we're all waiting for more information on this. So we've been working with a group of climate scientists to try to understand in a different way, focusing more on what the children might be breathing in, to understand what triggers this disease. And again, this is our climate team here. I'll point out Dan Pan, who's at Scripps Institution of Oceanography, so that's just down the hill from us. It is part of the University of California and one of the leading climate and oceanographic institutes in the world. And I'll point out also Jen Burney, who is a professor here at UCSD, also in climate sciences, and the work that she has done. Now, Jen was able to speak here last year. She's on sabbatical in France, and we couldn't figure out the time zone, so she's not going to be presenting, but I am presenting all her work here today. We like it that she's on sabbatical because she's actually had a lot more time to do this very intensive computer modeling of what's going on with Kawasaki disease. So what we did is we called out to collaborators across the world, and each place that you see a dot here represents in the data set date of onset of Kawasaki disease patients across the world. And Jen shared this with you last year, but I'll just go over briefly what her analysis involves. She's looking at the clustering of patients, because we think the clusters really are a key to what the trigger is. These children must have all been exposed at the same point in time to something that triggered their disease. And so what you see here is the time series for our patients here, cared for at Rady Children's Hospital. And everywhere where you see a black dot here is a case. And this is where there was 1 case per day, here there were 2 cases per day, and a few times there were 3 cases per day. You see this time series doesn't actually go up and include our amazing clusters that we have this winter. And when we look at this as an average, we can see that March is our really big month here, and then there's usually a little blip in the summer, and it's usually quieter this fall. That didn't happen to be the case this year, but in general, that's what happens. And we have to define what a cluster is going to be, and so they do a mathematical exercise where they take the same number of cases and have a computer tens of thousands of times simulate how many times would you expect to see 5 cases in a cluster. And a cluster is being defined as a period of 7 days. And how many times would you expect 3 cases or 10 cases? And the distribution then is plotted as these blue marks here, and then the actual data are the black marks. And we want to choose a definition of a cluster that is not something that could have happened by chance alone. So the Monte Carlo distribution is what could happen by chance alone, and we look at where the black dots are furthest away from the blue dots, and that's how we make our definition. So taking that definition, you can now analyze the data, how many times were there clusters, and then they do the climatology. So these are new data that are not published. And I want to walk you through this so you understand how these maps are made. First of all, you're looking at a map of the world here. And San Diego is a little hard for you to see, but San Diego's right here above Baja California. So that's our region that's being modeled. And imagine that, so let me explain the colors as well. Where you see green here, it's very low pressure, and where you see yellow merging into this orange color, it's very high pressure. Now, why do we care about pressures? These are called 700 millibar height potentials. But what that really means is high pressure and low pressure determines where the wind goes, and it determines what you feel on the ground, even though these are phenomena that are happening up in the troposphere way above us. But these pressure maps determine what happens with the weather, and importantly, what happens with the circulation, meaning the wind. So they then take every day where in this long 17-year period where there was no Kawasaki disease onset. And imagine that they can make the climatology for every day when there was no Kawasaki disease patients on a clear cellophane map. And then they stack all of these maps up. And so what you're doing here is you're looking at what we call a composite of all those dates, one on top of each other. And then here is a composite of KD cases that happened, their onset date occurred not within a cluster. So they're the non-cluster cases. And then these are all the clusters, and these are the clusters that happened during the winter period. So you don't have to be a climate scientist to get the colors here. And in addition to the colors, what I want you to look at are these black-gray dots. And these are the statistical significance dots. And where you see the gray overlying the pressures, either up or down, it means that this was a highly anomalous, very unusual condition that occurred at that time. So if we look at the days when there were no Kawasaki disease onsets, and we're looking obviously at San Diego because we're modeling the San Diego cases, nothing happening, right? It's a really boring picture. And these are a lot of days. These are thousands of days. There was nothing happening. When you look at the random cases that were just sporadic, one here, 2 weeks later we had another case, there's nothing happening. When you look at the clusters, you begin to see these black dots over our area, right? You see all those black dots? So it's high pressure because it's yellow, and the black dots mean it's a very, very unusual climate situation. So this is not happening by chance alone. There is something about the climate conditions, the wind, the circulation, that is exposing our children to this disease. So now we made contact with Naomi Kitano in the Wakayama Prefecture in Japan. She's an epidemiologist interested in Kawasaki disease research, and Chisato Shimizu in our group has been a wonderful liaison with our largely Japanese-speaking colleagues in Japan. And Naomi gave us all of her data, Jen modeled it, and here's what happens in Japan. So I drew a little circle for Japan because Japan is so tiny, it's very hard to see it, but that's Japan from here. And in Japan, there's a slightly different story, and that is that off of the coast of Japan, so nothing happening on no Kawasaki days, nothing happening here on random days, but clusters, which you saw in the last slides for Japan, there's a very, very strange climate phenomenon happening out here, and this is called the Aleutian Low. And basically what this is doing is it is sucking wind across Japan. So it's sucking wind from China, and it's flowing right across the Korean Peninsula and across into this sinkhole here, a very, very low pressure. So something climatically different, but again, something very, very anomalous happening. And now we have collaborators in the Southern Hemisphere. So this is Dr. Paul G. and his daughter, who just graduated from medical school, is now an intern. And they worked with the New Zealand Health Department. Dr. G is a Kawasaki parent, but also an emergency room physician in New Zealand. And we hope, Paul, that you're online joining us there. And Jen did the similar analysis of the New Zealand data. So you might imagine that New Zealand, everything in the Southern Hemisphere could be a mirror image of what happens here in the Northern Hemisphere. And in fact, that's true. That their high point is exactly opposite ours. Our high point for Kawasaki disease is opposite, it's their low point. So there's a mirror image, an exact mirror image of Kawasaki disease incidence in New Zealand compared to the Northern Hemisphere. So that, again, cannot be chance alone. And when Jen modeled, again, I have to put in a red circle so I can find New Zealand. It's very small out there in the South Pacific. But when Jen modeled their data, no Kawasaki disease patients, random Kawasaki disease patients. Here's New Zealand right here, so this is what you're focusing on. Nothing happening, boom. When you see the Kawasaki disease patients, this is actually during their vernal winter, so it's not December exactly, the vernal winter. You see, again, the high pressure. So it, in a place that's coastal, out in the middle of the ocean there, is behaving very much like San Diego with similar kinds of climate conditions. So I'm not a climate scientist. I did my best to share with you my understanding of what the climate scientists are doing, but the team is very excited. They think that this is going to lead us towards a better understanding of what the trigger is. These anomalous large-scale circulation patterns are clearly associated with clusters of Kawasaki disease. There must be an environmental exposure that is associated with these climate features that triggers KD in our genetically susceptible children. And here are all the projects that are now launched here at the Kawasaki Disease Research Center to try to take this to the next level. We have aerosol samples collected around the county that are being analyzed for their microbiome content, so what is in the air. Mike Levine in London is working with swabs from your children here in San Diego. So if you remember, we did a throat swab on your child when they came in through the emergency room after you signed consent. And those swabs have all gone to London, where at Oxford, all of the bugs that were on there, all the viruses, the fungus, the bugs, the bacteria, everything is being sequenced. And we have also febrile controls that will be prepared. Jen and Dan are continuing to work on climatology. We're doing antibody responses in our patients and looking at what antibodies they may be making to the microbes that we're finding from these different approaches. So I'm going to end it there, and I think it's now time for questions. We're running a little bit behind just because we started a little bit late, but if I could have the speakers come up here, and we will share the microphone, and we're hoping that we have people collecting the questions. Okay, if we could collect any questions that people have, or any questions from people online. So some of these questions we're going to, I'll be triaging them. We're going to save them if they are more relevant to the next session that we're going to have after the break. So we'll get to your questions, but we may answer them later rather than sooner. So there's a question about, it's sort of getting at how many of the children actually have lymph node inflammation. And while I showed you a picture of a patient that had a very large, obvious lymph node mass, we know from CT scans that have been done on other children that actually everybody has some lymph node reaction up in this region that would be filtering what's coming in through your upper airway. So it may not have been prominent in your child, it may not have been this giant node that we see. And there may be more than one trigger for Kawasaki disease, and maybe there's one trigger that is associated with the clusters that cause the big lymph node, and then there are other clusters where we don't see children with the big lymph nodes. So we're certainly pursuing that as a possibility.
So there's a question about whether Kawasaki disease remains a clinical diagnosis. At its heart, that exactly is what that is. We do actually have laboratories that can be evaluated. The American Heart Association diagnosis for KD includes labs for some children. And you may have heard our talks last year. Last year I presented some work that we're doing on diagnostic test development. So we have some computer programs that we're working on for diagnostic test development, as well as some other things working at the protein level and the RNA level, trying to figure out if we can find biomarkers for KD diagnosis. There was a question about the differential diagnosis of Kawasaki disease. This is probably what makes it so hard. It depends on what's being seen locally. I think that many children are diagnosed with presumed viral illness, probably the most common. Things like scarlet fever, in places where measles is endemic, measles here in San Diego as well. You can actually be misdiagnosed as having measles. So it is a very large list of things, and that's why kids can be confused with other illnesses.
So a question about the genetics of Kawasaki disease and have we seen patients with KD who then have children with KD? Yes, and in fact we published a paper about that, about pregnancy and Kawasaki disease. And we ascertained those pregnant mothers or those mothers when they actually brought their child in and would say things like, "You're not going to believe this, but I had Kawasaki disease when I was a child." And we go, duh. Yeah, it's a genetic disease. But unfortunately, there isn't widespread counseling and understanding of that. And so these moms had no idea that they could pass on the genetic pattern to their children. How often that happens, it's a complex pattern. It's not just one form of a gene that needs to be passed down. It's a very complex pattern. And we're working on that. And thank you to everyone for participating in our genetic study. Right now, just because of limitation of funding, we're only collecting DNA samples from people other than our new families coming group, if you've had aneurysms, but if you have had aneurysms, we're very anxious to have your DNA. So please consider contributing to our studies. It's a very active area of research. And another question about why do we wait to see the echo before wanting to initiate additional treatment? Why not intensify treatment for everybody? So we're always balancing cost and risk, and every treatment has a risk. There's no treatment that's risk-free. And every treatment, particularly the ones that we're testing, have a lot of costs. Not the statins, those are cheap, but anakinra and some of the other drugs are very, very expensive. So we're trying to target the people who really need it, and we think that our scoring system and the echo are probably a pretty good way of doing that. And at least at Rady Children's Hospital, you have your echo result within the first 24 hours of being diagnosed. Has the definition of aneurysm changed in the last 15 years? Yes. And the definition that I showed you with the Z-score handout up there, and we have that on the internet as well for you, has changed. So we have a more strict and more quantitative definition of what aneurysm is.
There's a question about flu shots, and when should a child that's had Kawasaki disease get their flu shot? My answer is yes, get it, definitely. Yesterday, get it. We don't want children to be exposed to flu and have the flu illness, which can be very devastating. And there's no reason to delay the shot, which is what's recommended for children because the intranasal, even though my kids, Mom, I don't want a shot. Well, guess what? The intranasal stuff doesn't work as well. So you're going to get your shot, and the shot, because it is modified, it's not alive, you can get that as soon as possible. There's no reason to wait, unlike some of the other like viral vaccines that we have to try to get. Another question that I'm holding has to do with long-term effects of KD related to other kinds of diseases. So KD mostly affects the heart. We don't think of any other significant long-term consequences. There's no neurological or developmental issues. There's questions here about recurrent illnesses or being at risk for other illnesses. And frankly, the answer is that gratefully our long-term work with the adult immunizations has shown significant links to most other illnesses. There are increased risks for eczema soon after, and that's for those that are part of our KD adult study. We sincerely appreciate your contribution to that. You're helping us answer those questions. But the specific questions that were asked about long-term consequences don't seem to be related to KD specifically.
Here's a climate question, and I'm just learning about the climate too. But wind never crosses the equator, FYI. And the other thing is that in terms of the hypotheses that we're testing, we think that there may be a source region in China where there are aerosols created that blow across Japan and trigger Kawasaki disease there. We don't necessarily think those same winds make it all the way over to the West Coast. We know from the Fukushima meltdown disaster that it takes about 4 days for that radioactivity to leave Japan and reach San Diego. So clearly we're connected through these currents of air. So it's possible, but we think it's probably more likely that there are aerosols being generated locally here in San Diego, and that high pressure cap is like putting a bowl on top of our region, and everything that's being aerosolized here is just trapped and circulating and can't get out, and it's something in that creates the exposure for our children.
So there's a question about triggers throughout the world, and specifically in Latin America. So we are working with our Latin American colleagues to collect the samples. Right now what we have is our data repository. That is all, you know, that was part of our research study. We've also collected blood and swab samples and other samples to be able to understand the cause of KD. That is just starting to be launched within the Latin American network to see what's going on there.
So this year we didn't feature the work that Dr. Tremblay has really been involved in with biomarkers and trying to make a diagnostic test for Kawasaki disease, but there's a question here about machine learning and artificial intelligence. And yes, we have one of our own, actually the brother of one of our KD patients who's up at USC working with the ex-Boston group up there, and are working with Dr. Tremblay and the group at Stanford, all different teams working to model, coming up with a biomarker and a way of diagnosing Kawasaki disease quickly and easily in the emergency room. So we're excited about where that's going to go in the next year. So stay tuned, come back next year, and we'll have more information about that.
There was a question about if you're concerned that your child may have had Kawasaki disease, but they weren't diagnosed with KD, what to do. I think this is a critically important question, not only for your children, but for family members. You may have, your kid had KD, you go to the Christmas party, and the aunt says, well, little Johnny over there had measles when he was 10, but if he peeled, and it sounds like it's KD. So we are here to help you find, if we can, I call it, we do our investigative reporting to see if maybe you had KD or your child did or a family member did. We're very blessed to work with an incredible team, also the cardiologist, who we'll meet again this afternoon in a little bit. And so if you're a child, you would come to us and we can see your child in our clinic and do a little Sherlock Holmes to see if it's a KD compatible illness. If you're an adult, then we can get you to the right adult cardiologist that, a couple of them are here in the room with us today. So let us know how we can be of help and come up with the right diagnosis. Yes, calcium scoring as well, yes. So the adult study that was done through your efforts and through being part of our studies has shown that a lot of fancy tests can be done in Kawasaki patients, but a calcium score is probably the most helpful test to be able to figure out if you would have had KD. So we have, for example, family members that they think maybe they had it, and we would get a calcium score that looks at calcium in the heart, in the arteries of the heart, and it helps us understand if maybe you had an aneurysm as a child that would have calcified over time. So it's a great, easy, cheap test that we can order for you guys.
A lot of questions about things that happen to children afterwards, like getting at the idea, do they have an abnormal immune system? Are they more prone to viruses? Are they more prone to ear infections? And these kinds of things. And we do not have a signal that our children are immunologically abnormal after Kawasaki disease. They obviously are doing something abnormal in response to this particular trigger, but not to other triggers in general. No questions about zebrafish or Dr. Madigan?
It was all so cool and clear, you just love to kind of, wow. Wonder what mine will look like firing it. Something was right? Yeah, how are children, you know this, Eric, how are children treated for KD before IVIG came in the picture? They weren't. And in fact, many still aren't, right? So many parts of the world, the family can't afford to pay for IVIG, they don't get treated. So that's part of our efforts, is to really understand what happens globally, and if we can be, better ambassadors for the world of children with Kawasaki disease to find better, more affordable treatments for those children.
So I think we're going to wrap it up there. We started a little bit late, but we know that part of this event is for all of you as parents getting together and talking to each other over a cup of coffee out there. So our wonderful lab team, research team has put together some refreshments out there. And so please stop by the table if you haven't already participated in our genetic study. If you're an adult who had Kawasaki disease, stay tuned, because the next session is really going to be a lot about outcomes and the future. And we'll get to some of those other questions when we have the question and answer period at the end. So don't despair if we didn't get to your question here. You can either ask it again or we'll get to it in the last question and answer period before. So why don't we all try to be back in here at 10:45, and thank you.
So electrocardiograms or EKGs, which I'm sure everyone is familiar with, that assesses the electrical activity of the heart, but that can also provide some information about damage to the heart. So if one of us goes into the emergency room right now and has chest pain, one of the first things they're going to do is do an electrocardiogram because that gives us some information about any potential damage to the heart muscle. We can do blood tests for heart muscle damage. You might have heard of a test called troponin, BNP. Troponin is protein that is inside heart muscles that when the heart muscle is damaged leaks out into the bloodstream. And so if you take some blood, you can tell if there's been heart damage from a heart attack or something similar. And then we can image the heart muscle. And so we can do that by different ways. There's echocardiography or ultrasound, MRI, CT, nuclear medicine scans. There's multiple different ways to image the myocardium and the heart muscle. They all have various advantages and disadvantages. There's not one that's perfect, and often, as I'm sure you've experienced, you get more than one for that reason. They often complement each other. So echocardiography or ultrasound is really nice because it's quick, it's safe, there's no radiation, it's fairly easy, it's painless, there's lots of great advantages to it. So one of the things we can see, when you see just the still frame image here, is this is a coronary artery in a certain view from the heart. And so what we can tell from this are things like where it is in the heart, how big it is, one can measure this. Is there an aneurysm there? Is there a narrowing? So we can get some characteristics. We can't get things like coronary calcium, so we have to do a CT scan to do something like that. You can see the artery pretty well, especially in children that have nice thin chest walls. Have really good pictures from ultrasound. You can see flow in the coronary arteries. Play this. Thank you. So you've probably seen this if you've ever had an ultrasound, but it's this red and blue color, which shows both the speed and the direction of blood flow in the heart. And this picture here is that artery that we just saw. Sorry, it seems a little jittery, but that's okay. You kind of get the idea. You can kind of see some flow in that artery. So we look and see if that flow looks normal or not. So ultrasound is able to do that too. So ultrasound has both of these aspects of those tests I talked about up front, which is looking at the artery itself, which I just showed you, but then also the ability to look at the heart muscle and myocardial movement. And so here is an image of the heart moving. So you've probably all seen something like this. You can see all four heart chambers here. Right atrium and the left atrium, and the right ventricle and the left ventricle. All of this down here is the heart muscle that's squeezing and propelling the blood out of the heart. And you can see the valves, two of the valves in the heart as well. And so, because there's no blood flow through those arteries to this heart muscle, we see that in this normal picture here, the heart muscle is moving perfectly normally. Okay, so the coronary arteries, most of us have two. And they go throughout the heart, and they have branches, and they reach every portion of the heart. So, sorry, it's a little bit faded here. You can see the light here, over here on your left side of the screen. There's a right coronary artery, which is shown in purple here, and a left one. And all these branches throughout end up penetrating all of the parts of the heart. And so if we know, for instance, that someone had an aneurysm in the right coronary artery and is at risk for having a blockage where that aneurysm was, something like here, then we know that that coronary artery corresponds to, that corresponds to a certain part of the heart, and we can look at that part of the heart when we look at something like this and look at that part and say, is that part moving like it should be? Is that part moving as well as the other parts that don't have any issues with their coronary arteries providing their blood flow. Okay, so we can do that, and we do that routinely, and we do that at rest. Now, there's an important aspect of exercise to this whole thing, and the reason why exercise is important is because it makes the heart work harder. And when the heart works harder, it needs more blood flow to do that. So if we go out and we make you run as hard as you can, so maximal effort, maximal exercise, parts of the heart, like the left ventricle, actually need up to 7 times the amount of blood that it does when it's at rest. And so, in order to allow that to happen, the coronary arteries relax or dilate and enlarge, and they can accommodate the increased blood flow that the heart muscle needs to do that extra work. And so abnormalities that are in the arteries that limit the flow may affect the heart muscle, and it may not affect it at rest, because if you just have a slight narrowing, the heart muscle can get enough blood flow at rest through that slight narrowing. When the heart muscle needs a lot more blood, like at exercise, then that narrowed artery might not be able to accommodate the amount of blood that the heart muscle needs. And so something that might not be apparent or significant at rest might become significant during exercise when the heart needs a lot more blood flow. And that's the concept behind doing exercise testing and imaging of the heart with exercise in these patients. So as I said, it may only be apparent in the high flow state of exercise. Okay, so we routinely do exercise stress testing. There's a guy on a bike who looks like he's getting ready to do it. All smiles, of course. It's very fun. And so we attach EKG leads. Everyone knows what an EKG is, but we have EKG throughout the tests. At least at Rady Children's Hospital and at most places for kids, we have both a treadmill and a bicycle. And the way it works is not a way that any of us would go work out in the gym, where we kind of pick a nice sort of level and kind of cruise through and run for a while. The way we do it is what we call sort of a ramp protocol. So you start out really easy, and over the course of about 10-15 minutes, the exercise just gets harder and harder and harder and harder and harder. And what we're trying to do is get you to all the way to sort of maximum effort, so that heart is pumping maximally by the end of 10-15 minutes. So that EKG is on there continuously, so we can see any heart rhythm abnormalities or any hints of heart muscle damage if the blood flow is not getting to the heart muscle adequately. We can get even more information sometimes. We put a mask on some patients, and we can monitor how much oxygen and carbon dioxide their bodies are using and producing. And we can do tests of the lung. There's a lot of things we can do during the exercise test. But for the purposes of Kawasaki disease, what we really want to know is what the heart muscle is doing. So in addition to the electrocardiogram, we'll do an ultrasound, just like I showed you in that previous picture, at rest. And we'll put someone on the bike or the treadmill, have them exercise all the way to a maximal effort. We'll throw them back or have themselves thrown back onto the bed. And immediately after, as soon as we possibly can after, we'll exercise them. We'll image them again immediately post-exercise. And so, sorry that it's a little herky-jerky on this, but I think you get the idea that on the picture on the left there at rest, that's a normal heart beating normally, and post-exercise it's beating really quickly, and it's also beating more vigorously. The way that our heart provides our body with that extra blood flow during exercise is really by 2 ways. One is by increasing our heart rate. So we might go to a heart rate of 60 at rest, up to 180 or 190, or kids well over 200 sometimes when they're going as hard as they can. So heart rate is one of the reasons, and the other way to do that is to squeeze harder with every beat. So there's lots more beats, and then there's more blood going out with every beat. So if we look at the heart after exercise, it should be squeezing even more vigorously than it is at rest. And if there's some limitation to blood flow in one of those arteries, then we'll more likely see a difference between the section of muscle that's not getting enough blood and those sections of muscle that are getting a normal amount of blood flow. So we call that an exercise stress echocardiogram. And we take multiple views of the heart, we look and compare the resting images to the exercise images. So if you have a nice normal post-exercise echocardiogram, that means even with all that effort and all that work that the heart is doing, there's plenty amount of blood that are going through the coronary arteries to get to the heart. So the advantages are that it's pretty fast. We can do that whole thing in about 45 minutes. There's no radiation. We supervise it so that it's a safe test. And it's relatively inexpensive compared to a lot of the other tests we do. There are alternatives to it. For children that can't exercise, we can give them medicine that sort of try and mimic the effects of exercise. It's not perfect and it can be uncomfortable. So we always try to do exercise if we can, but there are alternatives. And then there are different ways to image the heart, as I talked about up front. So not only ultrasound, but MRI and CT scan and nuclear medicine. All of these things, again, have advantages and disadvantages. If anyone's ever had an MRI, and you can imagine trying to exercise while you're getting an MRI, it's not easy, right? But it actually does exist. We have a way to do it. You put bicycle pedals at the end of the scanner, and while you're lying there and your heart is being imaged, you're pedaling lying flat. So it's not very natural, but there are ways to do it. So there are alternatives to this test. So as we move into the future, we still need lots of work and lots of research to really determine what the best tests are for assessing these things. Dr. Burns and Tremolay and I are always debating how to assess our patients for coronary abnormalities or those with coronary abnormalities and how important or significant they are. And so there's a lot of work and research to be done, and this is a great place to join it. But in terms of what we're doing right now in 2019, what I just talked about is probably what you and the other kids and our patients are doing. So I'll stick around for the Q&A.
Thank you, Chris. That should have looked familiar to many of you in the audience whose children have had those kinds of tests, but we thought it was important to have a review so now you actually understand what was happening when they had those tests. It's my pleasure now to welcome Cami Asaro, who I first met many, many years ago, and Cami agreed to come and talk with us today and to share her experience as a Kawasaki disease patient. We're absolutely delighted to have you here, Cami, and thank you so much for coming.
Can you guys hear me okay?
Yes.
Hello, my name is Cami Asaro, and I have been a patient with Dr. Gordon and Dr. Burns for the last 15, going on 16 years. My Kawasaki journey started in the spring of 2004. It was the second semester of my freshman year in college. I became really ill and stayed home from school for about a week, assuming that I had the flu. At about day 9, when my fever still has not subsided, my parents became really concerned. My mom and aunt decided to take me to the ER at Sharp Memorial Hospital. When we arrived at the ER, the intake nurse took one look at me and wheeled me straight into the ER. They took my temperature and proceeded to run some preliminary labs to try and figure out the cause of my fever. The labs came back negative for influenza, toxic shock syndrome, because I had pierced belly button and STD. When the labs came back negative, they decided to do a spinal tap to rule out meningitis. The results for that was also negative. After all the labs came back negative, I was admitted into the ICU. I was in the ICU for about a week, going on 2. Not sure how long I was there because I was in and out of consciousness. I had more labs drawn every single day that I was there with the hopes of finding the cause for this fever. Because no one knew what was wrong with me, I was also put on NPO, nothing by mouth, which meant I could not eat or drink in case I needed emergency surgery. All I was given were fluids via IV. After my extended stay in the ICU with no viable prospects of a diagnosis, the doctors discussed discharging me so that I could be more comfortable at home. The day before I was to be discharged, I had a new doctor doing rounds in the ICU. She was actually here today, Dr. Nancy College. Thank you. When she looked at me, something clicked in her head, and she basically said, you know, I just had lunch with a colleague the other day, and she was telling me about this heart condition she was researching called Kawasaki Disease. The symptoms that you are showing with prolonged fever, rashes all over your body, swollen lips, and strawberry tongue, it looks like you can have it, but here's the thing. You're too old to have it because this condition only affects children between the ages of 3 to 5, and you're clearly not 3 to 5. But I'm going to contact her over at Rady's Children's Hospital anyway to see if we can get some labs run for you over there to at least rule this out before we discharge you. So the next day, I was put on a wheelchair and wheeled over to Rady's Children's Hospital. I had an ultrasound done and a scope down my throat to capture images of my heart. Lo and behold, there on the left side of my coronary artery was a 4-millimeter aneurysm just hanging out. I finally have a diagnosis, and it was a huge relief to have a name to this invisible enemy. And for that, I'm forever grateful that Dr. Burns is much. I was immediately put on immunoglobulin IV treatment and given Lovenox injections. My persistent fever broke within hours. I was monitored a few days and was finally discharged. I began seeing Dr. Gordon at the San Diego Cardiac Center. I started to take Plavix and a baby aspirin to prevent a blood clot from forming in my aneurysm. To be honest, I wasn't always the best patient. Let's face it, at 18 you think you're invincible. So after a year of taking my meds, I assumed I was healed. Keyword. Because I thought I was healed, I also stopped seeing Dr. Gordon. Not the smartest decision, but who can say they're smart in their early years into adulthood, right? Dr. Gordon eventually tracked me down and explained to me the importance of anticoagulants for someone with my condition. It's a preventative for a stroke, heart attack, or even death. He also explained the new findings in which KD affects not just the arteries, but the heart muscle as well. And it is very important I stay on top of my health. So I came back to the cardiac clinic and went to see Dr. Khan to get an updated CT angio, since I had not had one in a few years. I can't remember the results for that one, but it had both. So because I'm a terrible patient and my aneurysm grew, I was put on Coumadin. Honestly, Coumadin has to be the worst medication ever. It is so restrictive and finicky, and I can never get my INR right. And the diet. You're telling me to refrain from dark leafy greens and vitamin K rich foods. I live in San Diego. All we have are trendy salad restaurants and green drink smoothies. Eventually, Dr. Gordon introduced me to Xarelto. Finally, an anticoagulant I could get on board with. In 2014, I met a man, and we fell in love. Right over there. 2015, I started a new job with excellent benefits, but the HMO required me to get medical care with someone else. That's okay. Dr. Burns and Dr. Gordon stayed in contact. In 2016, I married the man I fell in love with, and we wanted to start a family because, let's face it, nobody's getting any younger. With the HMO plan that I had, I went to see the cardiologist they assigned me to. He was a very nice doctor, but not very familiar with KD. First thing he did was order a stress echo and CT angio. My stress echo came back great. Happy, healthy heart. However, my CT angio revealed my aneurysm had once again grown. This time it's 8 millimeters and now considered large. Because the aneurysm is now large, he wasn't comfortable with giving me the green light to get pregnant. Even though I had advised him, Dr. Gordon is more than happy to discuss my treatment with him. And I provided him all the reports and research findings Dr. Burns had provided. He insisted we meet with the high-risk OB first to make sure we are making an informed decision and to prevent complications. I thought it was very reasonable and just made sense. So my husband and I met with the high-risk OB. The high-risk OB got straight to the point. Very matter-of-factly, she stated, I don't recommend pregnancy for someone with your condition. There just isn't enough about Katie to say that it's safe for you to go through pregnancy. And honestly, if you're pregnant, I suggest you get an abortion. You should really think about going the surrogacy route. When she left the exam room, I just sat and cried in my husband's arms. I felt so defeated and was utterly speechless. Moving forward with the high-risk OB's recommendation, we went to see the fertility specialist. Turns out they don't actually provide the hormone therapy and egg retrieval and implantation. They just offer guidance. For someone with my condition, according to the doctor we saw, I would need to find a private fertility clinic with their own anesthesiologist, because if I meet the anesthesiologist of the day of the egg retrieval, if he deems me as a liability from my chart, he has the right to not put me under, and all the hormone therapy would have been for naught. So my husband and I went home and discussed our options, and surrogacy, though an option, was extremely expensive, and I as a woman has always wanted to carry my baby. I was heartbroken the experience would be taken away from me. With a heavy heart, I sent an email to Dr. Burns to let her know I was giving up because I was just so tired of crying over a baby I cannot have. She replied back in caps lock, "You've been given the wrong information." With that, at the next open enrollment for my company, I signed up for a PPO and saw Dr. Gordon in the next year. With Dr. Gordon's guidance, I found a high-risk OB and perinatal specialist willing to work with me and my condition. We switched from Xarelto to twice-daily Livanose injections. In October 2018, we found out we were pregnant. But with a heavy heart, we had to announce our miscarriage just days before Thanksgiving. I was devastated, obviously, but my husband and I took two things from that experience. One, at least we know we can get pregnant. And two, it was just not our time. We obviously did not let that deter us because we are now expecting our little girl this winter. Life with Katie isn't scary, and you don't have to walk on eggshells. I live a very full and active lifestyle. I have a full-time job. I hike. Heck, I ski. I love traveling and exploring new places. I have an amazing husband who takes fantastic care of me, and the world's best cardiologist to monitor my heart. Honestly, if there's anything I can share with you all about my life with Katie thus far, is make sure you find the right doctors and advocate for yourself and your child. Everyone is afraid of the unknown, but if we continue to share with each other and those around us, Katie becomes a little bit more known, and that's a great thing.
Cami, that was an incredible testimony that covered a lot of ground, and I wish your story was unique about the misinformation that you were given. But thank you for sharing your story. It's my pleasure now to introduce Dr. John Gordon, who is the clinical director of our adult Kawasaki Disease research program, and who sees many of our patients who graduate from Rady Children's Hospital with important heart problems and then are cared for at the San Diego Cardiac Center. So Dr. Gordon's going to share with us today new advances that we're making in imaging the heart and how we can continue to monitor our patients and make sure we keep them safe. Thank you. Truth in advertising here. Dr. Gordon is also my husband. But we are a husband-wife team. And
thank you, Jane. And good morning to everyone here. As Jane said, I'm an adult cardiologist. Actually not an imager, but we're going to talk about imaging. Andy Kahn is not key here, so I'm going to give really his talk. Answer some questions about how we take care of adults with Kawasaki disease. I just want to thank Cammie for her kind words and sharing her story. It turns out that actually if you're cared for, if you're monitored, pregnancy is really not a big problem, and our patients do quite well. It's something that we did learn from our adult Kawasaki study and has been published in the OB literature. So let's move on to imaging of the heart. If you had no difficulties, if you had no aneurysms, no injury as a child, and that's been clearly documented, you're done with imaging, and all's going to go well. So no advanced imaging needed. But the patients who have aneurysms are in an entirely different category. We'll talk about a little bit about the spectrum of outcomes that can occur if you have aneurysms. Now, why do we do imaging? Chris gave a lovely talk about how we assess function. But basically, we're really trying to determine what the risk for blood clots might be. Are there valves in the artery design as well that should be addressed in some way? And we're interested in assessing the function. So, how does it go if you have an aneurysm as a 4-year-old or as a 4-month-old? And this slide summarizes at least 4 different ways. It can be, the arteries can remodel perfectly normal functionally. They can improve. Aneurysms persist. You can have aneurysms that don't grow, don't remodel. They stay basically the same. And unfortunately, there's some that enlarge and lead to complications. And this, instead of having weight slides, we're going to have pictures. This is a young man having an echo. This is the same fit. I think we use the same glue. Get the same happy child on his exercise bike, as you saw a little bit earlier. We've got somebody who looks more like my patient, a little bit older. But the idea there is to make the heart work. If there is a narrowing in the artery, we can see that the heart doesn't function properly, and then we can act on information. Dr. Kahn, who can't be here, is our CT angiography, and this is one of the machines that he uses to image the heart. And what it does is it spins very rapidly, and because of that, it uses a very small amount of radiation to image the heart in an extraordinary way. And we get these kinds of images. And this just illustrates, I guess the pointer's not really working. It is working? Okay. Very well. But if you look just to the left of the aorta, you can see that there's an aneurysm. And this is really what we're looking for. How big is it? Is there calcium? Is there thrombus? This is, we don't really have to do an invasive angiogram many times because we have this quality of image. And this just illustrates what it looks like to be inside one of these CT scanners. And what we're illustrating in the right panel are calcium that we can detect in coronary arteries. And this is, we learned from the adult Rady study that you begin to see a signal of coronary calcium that reflects aneurysm. So if you've had an aneurysm, just about everybody will get some calcification in the artery. And so if it's not known if there's important injury to the heart or aneurysms, this is an inexpensive terrific screening test that we use all the time to help either make a diagnosis or reassure some people who had Kawasaki disease that they don't have a problem. In this case, we did detect calcium. So this illustrates MRI, which we don't really need very often, but is another way of imaging the heart. And this is these images illustrate what we do when we do nuclear stress testing. Basically, we do, you can see in the top panels here, a resting study, and then we do stress. And if part of the heart muscle doesn't work, it drops out. This is over time. All right, so this is normal, this is not. This is normal, and this is not. So the heart muscle is not getting blood properly. The image is changing. That's how we make it up. Yeah. And this is the kind of procedure that we've traditionally done for coronary artery disease. Unfortunately, we don't have to do it as much in the pediatric patients. But if we want to intervene, this is how we gain access to the arteries and the heart muscle and the heart arteries. And we have terrific imaging, and then we can perform interventions. So, is heart muscle damaged after Kawasaki disease? And the question that Dr. Khan is, and his colleagues are trying to answer is, can we non-invasively answer this question? The panel on the left shows normal heart muscle, and on the right we see scarring. So at UCSD, there is a novel imaging technique that's been labeled or called SQUEEZE, which stands for stretch quantification for endocardial engraved zones. And basically, it's some complicated math analysis of a CT angiogram. And let's see if we can make this play. And this is what the CT image looks like on the right, and you can see a normally functioning left ventricle. On the left, you see the math that's being done on every little segment of the heart to get this kind of imaging on the right. And let me tell you one story. So this patient had Kawasaki disease stage 10 month with a delayed diagnosis, and presented ultimately with giant aneurysm of the left anterior descending artery. At age 23, suffered a mild stroke. This was, he behaved a little bit like Cammy. He went AWOL and wasn't being followed, wasn't being treated. And the SQUEEZE study shows, or illustrates what the problem was. What you can see here at the apex of the heart is part of the heart muscle which is bulging out instead of squeezing in. And blood can sit there and clot. And a blood clot formed there, broke off and went to his brain and clotted. And all of that can be prevented and treated, and that's what we're doing for him now. This fairly complicated slide shows how, with the squeeze study, the 17 segments of the heart are analyzed, and the part of the slide inside this blue box is the distribution within the artery that caused his heart attack, the left anterior descending artery. And if you look, what you can see is that this red line is above the black line, which is abnormal. And what this ultimately should be is a way of determining if there's fibrosis, if there's injury, and hopefully a more sensitive way of doing that. I'm going to tell you one second story. This patient had KD at 18 months, had a giant aneurysm in the left anterior descending artery, but the aneurysm remodeled by echo. She did well, and at age 35 had a squeeze study. And what you can see here is that her heart functions entirely normally. So giant aneurysm, remodeled, supplies the heart normally, no injury. The squeeze study shows really no evidence for injury or fibrosis. And this is a CT angiogram showing lovely arteries. The calcium score is 0. So this is one of our patients that remodeled very nicely, and it really has normal arteries and should be fine. So there's, as you've heard, there's ongoing research on many fronts. The squeeze study offers the promise of being able to find evidence for fibrosis or subtle damage to the heart. But we'll see. This slide summarizes some of the studies that are being done by the adult KD study. And genetic studies, biomarker studies, we're going to do a clinical trial that looks at these novel anticoagulants, Eliquis versus Coumadin, and their health questionnaire. The adult study has currently enrolled more than 425 subjects. We have 2 cohorts, one that came from Rady Children's Hospital, cohort 1, and we have a second cohort of patients who found us on the internet or have come from around the country to join our study. This is how we learn. This is how we learn that a CT coronary calcium score can detect aneurysms. This is how we learn how patients who are pregnant will do and why we can reassure them. So this is, we think, important work. And consequently, if you're 15 years or older and had KD, please participate. So we want you to participate in the research, and there's information outside of that. So that's what I have.
Thank you, Dr. Gordon. So we're now going to have the second and last question and answer period. So if I could have the speakers come up from the previous session. Cami, come on up. We'll let you sit down until you have a question that you need to answer. And if people can pass in their questions, and Beth, if you have questions for people coming up. So, one of our questions from this morning that we didn't answer, but it sort of fits into this category. Well, John's looking at the questions, was how long should patients be followed after Kawasaki disease? And I know if your children are being taken care of at other centers across the United States, there's a huge variation. And I just want to state that the American Heart Association guidelines give permission to stop following a Kawasaki disease patient after 6 weeks or so after the acute hospitalization. For those of you who come to get your care at Rady Children's Hospital, you know that that's not what we do. We feel that there's still a lot of uncertainty about this disease. If we're not collecting information about these children going forward, we're never going to learn whether that recommendation is actually a good one. And so we follow your children through to the teenage years. When they get to be 15, we invite them, and you have to give your permission as the parents to join our adult KD study, which is a questionnaire. Dr. Lori Daniels is the head of that effort. She's sitting actually with us at the back of the room here. And if there are questions about the questionnaire, I'm sure Lori would be happy to field those. But Dr. Daniels is constantly analyzing these data, and we are going to find out what the real answer is to the question of, as a Kawasaki disease patient, is there anything that may happen to me when I'm 20, 30, 40 years old? And if anyone tells you that we have the answer to that, please have them write me, because I'd like to hear how they know that. There isn't any data, and I can say that the adult KD study here, with funding from the Macklin Foundation, was the first of its kind in the world. And in fact, the Japanese, several years later, after our making presentations, Dr. Gordon making presentations in Japan, where he is invited back because he really follows the largest single group of Kawasaki disease patients with aneurysms of really anyone, that they have now just launched their own version of a longitudinal study of adults after Kawasaki disease. And we're delighted because there are over 150,000 adults with aneurysms in Japan who are not, up until this time, had not been followed or analyzed in any kind of systematic way. So, you ready for some questions?
Answer a couple ones here. One is it necessary to do a stress echo as a control after Kawasaki disease? I think you could speak to this in terms of what your practice is of when to refer a child for a stress echo. One, an obvious time is when we have known coronary artery issues, and we're trying to determine how significant those are. I think, Jane, you've been referring teenagers as sort of a, okay, you're 18 now, you're moving on, as a sort of goodbye. You might have had aneurysms at one point, but you haven't had any other problems. We're going to do a stress echo, kind of as a one more level of safety to look and see, are there any issues going out into adulthood before they go to Dr. Gordon, or before they go away, or hopefully not go away. So I think, is it necessary? Just what Jane said, we're trying to find out if that's the right thing to do. Again, tests like that are non-invasive and relatively painless, but where we can get good information is probably a good idea. One other question, probably for me, will KD kids have difficulties with exercise or physical activities after KD with only small heart issues? Again, depends on how small or what the issues are, but certainly by and large, and Cammie's can attest to that, most, if not all kids with proper treatment and proper examinations can live a normal life and not have exercise restrictions. That's another one of the reasons why we do stress testing. We feel better if you've had a normal stress test, you haven't had any problems during a maximal exercise test. We feel better sending you out onto the field or to the court to do various sports. Obviously, if you're on anticoagulation, that can affect certain sports where there's a risk for bleeding or trauma in contact sports. Beyond that, for the most part, children with KD that are properly treated can live very normal, active lives.
Thank you, Chris. Well, I have three parts here. Should a patient 2 years old with normal echoes have any follow-up with a cardiologist after discharge? And I think that the answer to the question is if it's truly established that the arteries are normal from the beginning, this child's fine. And sometimes we do see young adults who they were told they were fine, but it's not truly clear. That's when we do a coronary calcium CT scan. Costs $100, very low radiation, and it answers the question about whether or not there's damage. So it really depends on how clearly normal this 2-year-old is, say, 20 years later. But if you come to Rady Children's and you're certified normal, then you're normal, you're a normal person, and should live happily ever after. Second question is, healthy heart suggestions for adults that have KDs, kids with little or no heart issues. Well, be active, be happy, eat a balanced diet. I think there's really, if there are any issues, hypertension, diabetes, so forth and so on, those medical issues need to be addressed, but there's no really specific thing that needs to be done if there's little or no heart disease other than live a healthy life. The third question is, what can be done for a 49-year-old man with giant coronary aneurysms throughout his coronary arteries? And what this reflects is severe vascular disease. And there's a lot that can be done and should be done in terms of making blood vessels healthier. There are probably a number of other medical problems, probably diabetes, high cholesterol, hypertension, and so forth, that needs to be addressed. So this person needs to see a cardiologist.
And just to mention that we do have a program for out-of-town and remote patients who want to come for a single evaluation. We are constantly battling with ignorance among the adult cardiology population, that there's a lack of awareness of Kawasaki disease. They want to treat it like atherosclerosis, which is a typical kind of adult heart disease. It is not that beast. It requires a very different approach. The medications that need to be used are different. And those people need to have genetic counseling and lots of things that adult cardiologists just aren't aware of. So we're trying to get the word out there, but there is a lot of misinformation out there. And I think, Cammie, your presentation was very poignant in that regard, that we just have to battle against a lot of misinformation or lack of information about this disease. There's a question about funding. Someone wanted to know where our funds come from. And our funds come from you. Everyone sitting in this audience can participate in our research by donating to our Kawasaki Disease Research Center through the Rady Children's Hospital Foundation. We have NIH grants. Some of our support comes from competitive grants from the FDA for some of our new drugs from the NIH. But we are largely supported by philanthropy. This is an orphan disease. This is not on the list of the top things that your tax dollar is going to in terms of the NIH budget. We're competing with asthma and diabetes and things that affect millions and millions of Americans. So we're an orphan disease, and philanthropy is the single most important way that we are currently doing our research and that we will stay alive in the future and not close our doors. So to all of you who have given, thank you for your support. There's a question about whether we get money from other countries. We do have a wonderful family who has been very generous with their support in the Philippines. But beyond that, no, it's all domestic, and it's because of the wonderful parents who have come to our clinic. So thank you. There's a question about children having flares of Kawasaki disease after being treated with IVIG. They can have recurrent fever during that immediate period. It's their bodies telling us that they need more treatment. But beyond that, we see a very low recurrence rate. It's about 9% in our Filipino population, again, based on genetics. It's extremely rare in European descent or Latino families, so virtually never happens. It is possible, however, to pass on the genetics to the next generation. Someone wanted to know when IVIG was introduced as treatment. I enrolled the first patient in the national NIH-sponsored trial on Valentine's Day 1984. In 1986, in January, it was the lead article in the New England Journal of Medicine. And later that same year, the American Academy of Pediatrics endorsed it and recommended it as standard treatment for all Kawasaki disease patients.
So I have a question about whether statins are a good idea in adults with Kawasaki disease who may have normal or close to normal lipid levels. And I think that the answer to that is yes. Statins turn out to do more than just lower bad cholesterol. It's an anti-inflammatory drug that has really changed the way we take care of adult coronary atherosclerotic disease. But there is more and more evidence that it's a good thing in Kawasaki disease as well. We're studying ongoing or evidence for ongoing inflammation in patients with Kawasaki disease. So if you have certainly aneurysms then it doesn't matter what your cholesterol level is. A statin, I think, is a good idea.
There's a question about follow-up for a child who had dilated coronary arteries. So remember the Z-score handout that I put up there. We consider an artery to be dilated if it's between 2 and 2.5 standard deviations above the mean normalized for that child's body surface area. So if a child is in that category, we've been sending them, based on the results of our adult study, for when they get to be teenagers, so 15 onwards, we've been sending them for CT calcium scores just in case to learn if there's any evidence of scarring. We're soon going to be ready to analyze those data. We have not found any problems in that group of patients with transiently dilated arteries. And once we analyze those data, we'll want to write that up. But right now, my sense is that no, there aren't going to be any problems or evidence of scarring later on, and follow-up with an adult cardiologist would not be indicated for that person. I think we've done it. All right. So thank you to all of our speakers for coming today. And if I could have Amelia and Nipa and Chisato come up to the front. I just want to thank them. These are members of our research team. And although this seems like a pretty simple event to make it happen, it actually involves a huge amount of work by all the people at the Research Center. And I really want to thank the three of them for the major, everybody contributed, okay? But please come on up and let's have a round of applause for Nipa, Amelia, and Chisato. Thank you so much for all your work. We really, really appreciate it. None of this happens without you. So these three lovely ladies and everyone else too. Thank you. All right, and we'll be outside if there are more questions. We'd love to chat and probably some more refreshments. Thank you so much for coming. Come again next year, bring your friends and family. We do something different every year to make this an event that you can continue to come back to and learn about Kawasaki disease. So again, thank you for participating, and we'll look forward to seeing you outside.